Welcome at Basics of Biology.
In this video, we will discuss a review article which is on phosphatidylinositol 3 kinase inhibitors as a new weapon to combat cancer. As you all know, phosphatidylinositol 3 kinase is a group of plasma membrane associated lipid kinase consisting of two subunits, which is P85 regulatory subunit and P110, that's a catalytic subunit. The role of regulatory subunit is to stabilize the catalytic subunit. Phosphatidylinositol 3 kinase is a family of enzymes that are involved in cellular functions such as cell growth, proliferation, differentiation, motility, survival, and intracellular trafficking. So the PI3 kinase mTOR signaling pathway is involved in various vital functions including cell proliferation, survival, metastasis, metabolism, and angiogenesis. Thus, many diseases arise as a result of dysregulation or mutation of PI3 kinase mTOR pathway, such as chronic allergy and inflammation, cardiovascular diseases, and cancer. About 30 to 50% of human tumors are likely due to the activated PI3 mTOR pathway. In mammals, PI3 kinase belong to lipid kinase families which phosphorylate the hydroxyl moiety of the inositol ring, yielding products of which the most characterized is phosphatidylinositol 3,4,5 trisphosphate, PIP3, the second messenger that activates another protein kinase B, or other cellular messengers such as a mammalian target of rapamycin, mTOR. PI3 kinase can be divided into three main classes: class 1, class 2, and class 3, depending on their structure and substrate specificities. Class 1A PI3 kinases is divided into two main subclasses, class 1A and 1B, according to their mode of regulation. Class 1A PI3 kinases are heterodimer that consists of P110 catalytic subunits and P85 regulatory subunit. The catalytic subunit of class 1A has three different isoforms, which are P110 alpha, beta, and delta catalytic subunits produced from different genes. P110 gamma represents the only catalytic subunit of class 1B. Class 1A enzyme can be activated through tyrosine kinase coupled receptors, while the class 1B are activated through G protein coupled receptors. Class 2 PI3 kinase contains three isoforms: PI3 kinase C2 alpha, PI3 kinase C2 beta, and PI3 kinase C2 gamma. These isoforms are considered as an example of monomeric lipid kinases because they lack their regulatory subunit. The single class third PI3 kinase VPS34 vacuolar protein sorting 34 is an important and have vital roles in autophagy, endocytosis, phagocytosis, and intracellular trafficking. Some studies consider mTOR as class 4 of PI3 kinase related kinase. It has a catalytic domain as well as rapamycin binding domain and C-terminal fat domain. mTOR composes of two functionally distinct complexes, mTOR C1 and mTOR C2. mTOR C1 is responsible for potentiated protein translation and cell growth through activation of several downstream effectors, while mTOR C2 phosphorylates AKT, so it regulates the signaling pathway. Overall, mTOR activity promotes accelerated growth and cell survival, both physiologically and in diverse pathologies including cancer and other diseases like metabolic disorders, neurodegeneration, and aging. So the PI3 kinase AKT-mTOR signaling pathway is involved in various vital cellular processes. So many diseases arise as a result of dysregulation or mutation of PI3 kinase AKT-mTOR pathway, especially cancer. Thus, it is really important to study this pathway to be aware with the most fruitful way of cancer control. The inhibitors of this pathway are considered as a fruitful strategy to combat cancer, and these inhibitors are classified according to the selectivity to three main classes, which are pan-PI3 kinase inhibitors, and the second is selective PI3 kinase isoform inhibitors, and the third is dual PI3 kinase mTOR inhibitors. Coming to the first, pan-PI3 kinase inhibitors. Pan-PI3 kinase inhibitors are ATP competitive inhibitors that target all four isoforms of PI3 kinase class 1. And the examples of pan-PI3 kinase inhibitors are buprolizib. It inhibits all four isoforms of PI3 kinase. And the second is PX866. It is a semi-synthetic potent analog of warfarin. It inhibits only three isoforms of PI3 kinase, which are P110 alpha, delta, and gamma. And the third is XL147. It is also against all four isoforms of PI3 kinase and shows weak effectiveness against mTOR. And the fourth is picticillib. It is also against all four isoforms of PI3 kinase and weakly inhibits mTOR. The second type of PI3 kinase inhibitors are selective PI3 kinase isoform inhibitors. These are considered as a new strategy to combat certain type of cancers depending on which isoform is amplified or mutated. It can be classified into four classes. First is selective PI3 kinase alpha inhibitors. This type of inhibitors can be classified into three main subclasses, which are morpholine-based heterocyclic PI3 kinase alpha inhibitors. Second is morpholine-based monoheterocycles PI3 kinase alpha inhibitors, and non-morpholine based PI3 kinase alpha inhibitors. The second is selective PI3 kinase beta inhibitors. They have a significant importance in PTEN deficient cancer type, especially in prostate cancer. And the third is selective PI3 kinase gamma inhibitors. As a variety of diseases related to influx of inflammatory effector cells, inhibitors could be treated, including inflammation, respiratory and metabolic disorders, and cancer. And the fourth is selective PI3 kinase delta inhibitors, as they are used as an effective target for the treatment of immune diseases as hematological malignancies such as chronic lymphoid leukemia and autoimmune diseases like rheumatoid arthritis. In this slide, I have enlisted some of the examples of selective PI3 kinase inhibitors, which are tasilisib, alpelisib, and TAG117. These are the examples of PI3 kinase alpha inhibitors. GSK 2636771 is an example of PI3 kinase beta inhibitors, and idelalisib and linolisib is an example of PI3 kinase delta inhibitor, and tenelilisib AS605240 is an example of PI3 kinase gamma inhibitors. The third type of PI3 kinase inhibitors are dual PI3 kinase mTOR inhibitors, as some of the inhibitors show mTOR inhibitory activity due to their binding to catalytic domain of mTOR, which shares high degree of similarity sequence homology with P110 catalytic subunit of class 1 PI3 kinases. Dual PI3 kinase mTOR inhibitors have better efficacy and they have overcome the resistance problem as well. Among PI3 kinase mTOR dual inhibitors, there are 7 agents in clinical trials currently, such as GSK 2126458, XL765, GDC, PKI, PF, BEZ, and BGT. Fortunately, GSK, BEZ, and BGT displayed remarkable potency in vitro and in vivo. So the combination of PI3 kinase with other enzymes is a successful anti-cancer strategy to overcome the acquired resistance to PI3 kinase inhibitors. This was all about the PI3 kinase inhibitors. Thank you for your kind attention.